Hello, everybody. My name is Sam Young. I use they, them pronouns, and I'm a rising biochemistry major at Amherst College. This summer, I worked closely with my mentor, Nathan Sintomadonic, in Miguel Pineras' lab on elucidating the function of OS-Sulfur 3,6 through seed physiology and mineral nutrition. Rice is one of the world's most important crops and is essential for feeding the growing human population. While there are several prominent rice diseases, rice leaf streak has become a growing threat as more productive rice varieties are cultivated, have been found to have greater sensitivity to the bacteria that causes it. Here you can see the streaks that are caused by rice leaf streak. They create large, wet, necrotic lesions that can reduce crop yields up to 30%. Leaf streak is caused by a bacteria called Xanthomonas oryzae, or XOC. XOC is able to use transcription activator-like effectors, or TAL effectors, to upregulate specific genes in their host plants for their own benefit. Our collaborators in the Bogdanov lab at Cornell identified that XOC upregulates a rice gene called sulfate transporter 3,6, or OS-Sulfur 3,6. While we know that XOC targets this particular gene, and the alteration of this gene expression could have the potential to mitigate the damage of bacterial leaf streak. However, until we know more about SULT 3,6's function in rice, we cannot truly understand why XOC targets a specific transporter. In order to characterize SULT 3,6, it's important to have a background of the SULT gene family. The SULTer gene family encodes for membrane transport proteins and is also composed of several subgroups. SULT3 is the largest subgroup, but is also the least characterized in the literature. True to their name, many of the proteins encoded by the SULTer family transport sulfate ions, although recent studies have also seen that some members are able to transport phosphate ions. Like I said, OS-Sulfur 3,6 is not very well documented in the scientific literature, and its current function is unknown. Our collaborators in the Bogdanov lab, however, created CRISPR-edited rice with a full coding sequence deletion of the Sulfur 3,6 gene. My goal this summer was to phenotype the differences between the wild type and mutant lines to elucidate the function of OS-Sulfur 3,6. So when we initially observed the wild type and Sulfur 3,6 knockouts, we saw absolutely no differences in the actual phenotype. If you were to look at the two plants in the greenhouse side by side, you wouldn't be any of the riser asking which one is actually the mutant knockout. However, preliminary germination observations of the SULT 3,6 mutant knockouts showed a huge drop in germination rates. Seeds of the mutant genotype had reduced germination rates of approximately 30%. In this plot, you can see our germination assay with day on the x-axis and germination rate on the y-axis. Our wild type was in red, as you can see, gets up towards 100% germination rates, but our two mutant genotypes, you can see the actual difference in germination here. So this germination assay gave us our only clue. What can we learn about SULT 3,6 based on these germination defects? And how can changes in expression of SULT 3,6 affect germination rates? In order to answer these questions, this requires understanding what substrate SULT 3,6 predominantly transports. So previous studies on a different phosphate membrane transport protein observed that when the transporter was disrupted, the resulting high inorganic phosphate levels reduced the size of seed starch reserves and disrupted the grain filling process. You can see here in this case, the transporter was considered recessive and it has lower starch content. And in the full transporter knockout, you'll see the starch has a really strange looking granules instead of the usual crystalline structure found in the wild type. Smaller starch reserves have a negative fitness impact on maturing seeds, and this could be the cause of reduced germination rates. On the other hand, if SULT 3,6 encodes for a phosphate transporter, we can look back at previous studies that show that sulfate is required for the biosynthesis of AVA, a plant hormone involved in regulating seed dormancy. Here you can see that if you have increased sulfate ions, this can lead to a cascade down the pathway, which will eventually lead in higher abscisic acid levels. It has also been shown previously in the literature that buildup of excess abscisic acid does cause reduced germination rates. These observations led to our experimental hypothesis. Embryo development, or seed loading, is perturbed during maturation in the Sulfur 3,6 knockout seeds. Our hypothesis has two parts based on the substrate transported. If this gene encodes for a phosphate transport protein, the mutant seeds will have increased inorganic phosphorus nutrition, leading to decreased starch accumulation. And instead, if this gene encodes for a sulfate transporter, the mutant seeds will have increased sulfate nutrition, leading to increased abscisic acid levels. Our first experiments will start by addressing hypothesis 1B. So like I mentioned before, we know that abscisic acid inhibits plant growth and seed germination, but we also know that gibberellic acid antagonizes abscisic acid and instead promotes seed germination. If the addition of GA3 can rescue the deleterious effects of the Sulfur 3,6 genotype, then we'd also expect to see higher levels of elemental sulfur and sulfate in the mutant seeds. So this plot shows a replication of the previous germination assay. The only difference is that our two mutant lines are instead joined together under one, Sulfur 3,6 for ease of view. When performing the same germination assay with GA3 added, you'll see that there was no significant improvement in the germination rates. There was actually no improvement at all in the germination rates. So this led us to believe that gibberellic acid does not rescue the mutant phenotype. And interestingly, the wild type and mutant seeds reach their max germination rate at the same time. As you can see, it doesn't really increase much from here. This shows evidence for seed inviability as opposed to just a germination delay. So even though GA3 failed to rescue the Sulfur 3,6 phenotype, we wanted to confirm the levels of both elemental sulfur and sulfate ions in the wild type and mutant seeds. It's especially important to measure both elemental sulfur and sulfate ion content because elemental sulfur is going to quantify all the sulfur actually found in the seeds. So that includes sulfur locked up in cell structures like proteins and organelles. Whereas if you measure the sulfate ion content, it refers to the amount of free sulfate usable by the seed itself. Elemental sulfur was measured using inductively coupled plasma, or ICP, and sulfate ion content was measured using ion chromatography, or IC. Here you'll see our wild type is labeled in blue, our mutants are labeled in red, and on the y-axis, you'll see the elemental sulfur content measured in milligrams per gram. Our second graph is very similar, except the sulfate ion content is measured in parts per billion. After running our t-test, we saw there is no significant differences in the elemental sulfur or sulfate ions in the two seed lines. This supported the results of our initial germination assays and leads us to believe that Sulfur 3,6 likely does not encode for a sulfate membrane transporter. We then went on to measure both elemental phosphorus and inorganic phosphate in the wild type and mutant seeds. We measured both of these for the same reason that we measured both elemental sulfur and sulfate ions, because a lot of the elemental phosphorus is actually bound up in things like nucleic acids. Similar to the previous graphs, elemental phosphorus was measured using ICP, and inorganic phosphate was measured using IC. Here you can see we actually did have a significant difference between the phosphorus and inorganic phosphate content of our wild type and mutant seeds. There appears to be a lot of variation here, but this is likely because we had to run our IC analysis one extra time, as not all the samples got analyzed from the first way through. This also supports the results of our initial germination assay, which show that the seeds likely do not have an increased amount of sulfate. However, we want to complete our phosphorus story. It's important that we also measure the phytate levels. Seeds store a lot of their phosphate ions in storage molecules called phytate, and as such, phytate levels serve as another important measurement of phosphate ion levels. With the help of Jason Weisinger in the Gland lab, we were able to run an assay to assess phytate levels. While this data is still preliminary and our sample size is a little bit small, you can still see a trend that our mutant lines have higher elevated phytic acid contents than our wild type lines do. This data, compared with our ICP and IC data, makes it appear that Sulfur 3,6 likely encodes for a phosphate transport protein. If you recall, part of our hypothesis stated that increased phosphate levels would likely lead to a decreased starch accumulation. If high phosphorus does indeed lead to seeds having decreased starch levels, we would expect to see morphological differences between the wild type and mutant seeds, as well as differences in overall starch content. However, we saw no significant differences in the appearance of our seeds, where you can see the wild type up top and the mutant up down below. Just like when we saw the actual plants, if you put these two seeds side by side, you see no differences. Also, interestingly, in our 100 grain weight measurements, or 100 grain seed weight measurements, we also saw no significant differences. This is particularly interesting because a decrease in starch reserves is also heavily correlated to lower seed weight, since starch is the main compound that's contributing to seed weight overall. So while we saw no morphological differences between the seeds, we also just wanted to confirm the total starch content in both the wild type and mutant seeds. We calculate this total starch content using Megazyme's total starch kit. While you do see quite a bit of variation between our two mutant lines, this variation is not consistent in either direction, and is likely just chalked up to general starch variation between the seeds and plants. To recap, compared to our wild type, the delta SOLT3-6 mutants had higher phosphorus, phosphate ion, and phytate measurements. And our current data shows no significant differences in sulfur, sulfate, or starch content between the wild type and mutant seeds, leading us to believe that OS SOLT3-6 likely encodes for a phosphate membrane transport protein. So these elevated phosphate levels do align with one of our hypotheses, but why wasn't starch altered in the mutant seeds? When we look back in the literature, we saw there may be an answer in an unrelated phosphate transporter. In a study done by Lan et al., in Arabidopsis, they found that when a particular phosphate transport protein was perturbed, there were higher phosphate levels found in the plant's reproductive organs. Additionally, these higher levels of phosphate actually resulted in the creation of reproductive organs that had stunted development and led to decreased germination rates in mutant plants. Here you can see the two wild types, and you see the seed pods for Arabidopsis and the pollen tubes. Whereas on the other side, in the actual mutant knockout, the seed pods are much smaller, the seeds inside are less defined. In the pollen tubes, their links are also heavily stunted. This is very similar to what we were currently seeing in OS SOLT3-6 and could likely be the cause of the germination defect that we saw initially. However, more experiments will have to be done before we can absolutely be sure that this could be a similar cause to what's happening in OS SOLT3-6. I'd like to thank my mentor, Nathan, our lab manager, Eric, and my PI, Miguel, for all the support and assistance I received this summer. My time here would not have been the same if I was placed in any other lab. I would also like to thank our collaborators in the Bogdanov lab, Johnny, Kat, and Anna Bogdanov, as well as Jason Weisinger from the Gland lab. Lastly, I'd like to thank Megan, Olivia, and George for all the work they've done to organize and facilitate the BTI RRU program, as well as our funders. If you have any questions, you can find me after the talks are over, or you can reach me at my email here, syoung23@amherst.edu. Thank you.